Hi. Today I'm going to talk about super resolution microscopy. So to give you an idea of where we're going with this is a confocal image of a Golgi, cis and trans Golgi, regular confocal microscopy. And then this is what you can get out of a STED microscope. So you can see considerably finer detail down at fractions of a micron. That's where we're sort of going with super resolution. In terms of spatial resolution of the optical microscope and biological specimens, what we can really, we've got these fluorescent techniques, wide field fluorescence, we talked about last week, confocal, and some variations on a theme. There's a whole class of variations that we sort of lump together as super resolution. So what they're really doing is beating the standard diffraction limit of imaging, so the optical resolution limit from a normal microscope, something like 200 to 250 nanometers, so sort of the order of half the wavelength. And that gets you down to sort of, you can sort of see some structure in bacteria, but not very much. But there's lots of biological things below there, and this is normally reserved for the domain of electron microscopy. So super resolution microscopy is interesting because we're trying to push down with resolution, but maintaining the real advantages, particularly of fluorescence microscopy, which is of specificity. So we know what molecules or thing we're actually looking at. So last week we talked a bit about image formation in the microscope and the work of Abbe. He has this formula. The resolution is basically proportional to the wavelengths divided by the numerical aperture of the lens you're using, so lambda over 2Na. His sort of argument, just to recap that, was that based on the work of Young, we sort of knew that light was a wave, like was a wave type phenomenon. Abbe sort of abstracted the object on the microscope down to this sort of, as if it was some form of diffraction grating. So the fine structure of your object diffracts light. And with that argument, he basically said, the finer the detail, the larger the diffraction angle. And in fact, so the information about fine detail is carried on these fine diffraction orders that have come out at large angles. If those don't get through the optical system, they don't get into the image. So you've lost. There's a limit to the resolution. Oh my God. How did that come there?
God knows what it's doing. Yeah, let's go back to the presenter.
Did I hit some button? All right. What that sort of means, if you have a point-like object, like a single fluorescent molecule, and you image it, you get this sort of distribution in space, and really the width of that thing is this resolution limit, so sort of the order of 200 nanometers or so. There's a different way of expressing this sort of idea, and you can just sort of switch that into a frequency domain, where you just sort of effectively Fourier transform this. And this is called the optical transfer function. So it's really just a convenient way of representing how high frequency, so fine details, pass through the microscope. And the idea is that the fatter that distribution is, the finer the frequencies that get through before it reaches zero. So there's no actual transmission down here, maximum transmission up here. So that is what Abbe's model of the microscope shows us about the optical transfer function. And what it's characterized by here is this hard cutoff at a frequency defined by the inverse of the resolution, so 2Na divided by lambda. So beyond that point, there is no high frequency information through the microscope. It's resolution limited, but it's got a hard cutoff. Is the point to note here. What does that mean? Yeah, again, we're sort of down at subcellular organelles, and then beyond that, we can't really see things. So the question is, super resolution are really the techniques that take us beyond that looking at macromolecular complexes, and you know, on a sunny day, maybe all the way down to single molecules. Conceptually, there is a limit to this thing. So we sort of start at the diffraction limit and get smaller. You can imagine that we shrink the finest detail down and down and down until at some point there's just one fluorescent molecule there. That point, when we reach that point, something else kicks in. The resolution is then determined by how many photons we get. If we've got one photon out of that molecule, we really wouldn't be able to say where it was with any certainty. And we'll get into that towards the end of this talk. But that is the sort of ultimate limit of super resolution. In the lab here, we're sort of down to 10 nanometers for single molecule localization. There are instruments around nowadays that will get you down to 3 nanometers to localize a single molecule, but that's a rather specialized device. Which now brings me on to the actual super resolution microscopy techniques. So they sort of come into 3 broad camps. The first one we'll talk about is structured illumination, where basically you put some sort of pattern of illumination on your sample and use that to extract some higher frequency detail, some finer detail. Stimulated emission depletion microscopy, or STED, which reshapes the point spread function, and then localization microscopy, which basically builds the image up fluorescent molecule by fluorescent molecule. So rather than seeing everything all at once, we see a small subset of the image spread out in time, and then we synthesize an image from that at the end. Structured illumination. So this, the idea for this is not new. We've got a paper here in 1967 from McCutcheon. So he was talking about the work of Geraldo di Francia, who said that you know, super resolution, that means resolution of detail finer than the Abbe resolution limit, is possible, providing that one has some prior information. And then if we skip down to here, this effect can be achieved by confining the illumination to a limited area. So this is the concept of structured illumination. I mean, I think, you know, historically, it didn't really occur to Abbe because, frankly, we didn't have a point scanning confocal microscope in 1860 or something like that. But this is the idea of structured illumination. You can illuminate a single point, and that's what we're doing in a confocal microscope. You can illuminate it with a grating, and we'll see the advantage of that shortly, or you can have multiple points. But we're not exciting the whole of the field of view all at once, which is what Abbe's model was considering. I don't want to get too bogged down in this one. Here we go through a few examples of structured illumination. Up the top is wide field. Then we've got confocal microscope, the single point illumination. Confocal microscopy with a shut down pinhole on the detector side. And then SIM, structured illumination microscopy, SR, which is really this idea of using a grating as the object. We go all the way to the end here and look at the effective point spread function, that red curve. You can see that sort of triangle from Abbe with the hard cutoff. We go into confocal, that function changes shape and now becomes a Gaussian. So it's got a smoother decay down here at that hard cutoff. We shut the pinhole down, it gets broader, so we can see a bit more high frequency information. So here is where the super resolution business is coming. We're getting amplitude in the optical transfer function beyond the normal Abbe cutoff. And then with the structured illumination, the grating illumination pattern, we can take it out by a factor of 2 before it decays all the way down to 0. So the problem with the confocal is, sure, as you shut the pinhole down, resolution gets finer. So we get down to sort of 1/√2 detail compared to an open pinhole. But the amount of energy that gets through that pinhole plummets So, we increase resolution, we get no signal. And so that's really a very poor microscope. So resolution improved at the expense of sensitivity. So that's not really a good idea. So we're always operating our microscopes here with a one area pinhole to try and get signal through to the detector. That's what that looks like. Wide field is here. Ooh, I've lost a slide. This is sort of regular wide field imaging of a stain cell, so TOM20, a mitochondrial stain, and a Golgi stain. We can see the problem in wide field, all this out of focus fluorescence above and below the focal plane. We go into this method called Zeiss-Airy scan, which fixes the issue of the pinhole. We get a lot more detail. I'm just going to skip out of here because I'm sure there is a missing slide somewhere. Ah, here it is. Let me just, maybe I just hit the button twice. All right. So we just talked about this business of shutting the pinhole and resolution and why closing it down. So there is a neat solution to that problem, and that is this so-called Airy scan detector. So this is one of a number of variations of a theme, but it sort of explains the idea. So in the microscope, we could either go through the standard pinhole up here, that one, or we could sort of basically magnify the spot and spread it out over a multi-array detector. And then each one of these detector elements is like a small pinhole. But we collect all the signal because we've got lots and lots of small pinholes and they're all shifted around in an array. So that gets us that amount of image with that amount of resolution. So that solves that problem, and that's what the Airy scan does for us. And that's the improvement we get. We do have another variation on a theme, which is the Nikon ISIM, which is basically similar performance. So we're already improving the resolution in the image. Now, if we consider this more extreme case, where we're doing the patterned illumination with a grating, that's that sort of illumination on the sample, we can push that resolution advantage even more. So we do have one of these instruments called the Elira S1. That's what it looks like. Just round the back of the microscope is a device for putting the pattern onto the sample. We're in sort of regular imaging in many respects, so we still think about wavelength as a factor in resolution. So, blue dyes with shorter wavelengths give you higher resolution images than far red dyes. But the instrument itself, interestingly, it's still a regular microscope. It's just got a pattern in the illumination. And how it works is this. So here's a picture from some zoo or other. This is a cage where you've got a grid-like sort of barrier at the front of the cage and another one at the back. And when you look through the both of them, this pattern here, the grid of the one, interferes with the other, and you start to see these sort of darker lines. So that's basically a form of mixing. We're basically just looking at one grid through another grid, and we end up generating this low frequency information here, these dark lines. So the idea would be, if I knew what the frequency of, say, this grid here is, the one I can see at the front of the cage, and I know the periodicity of this low frequency pattern here, I can work out what the frequency of the grid at the back of the cage is. So the information about that grid at the back of the cage is carried in this low frequency pattern. And that's the trick for taking high frequency information out of your sample and transporting it through the microscope. So this stuff because it's low frequency, can get through the microscope. And that's sort of the gist of how it works. I didn't see, yeah, so here's a sort of another version of it. We've got a grid here and a pattern here. And if you sort of tilt them one relative to the other, you can start to see that these other darkish lines come up. That one doesn't work very well. All right. So just to recap that, our sample, the unknown, has got fine detail in it that in principle doesn't get through the microscope because it's beyond the frequency cutoff. We illuminate it with a slightly lower frequency pattern. Those two beat with each other. This is called heterodyning in sort of frequency in signal processing, and it generates lower frequency information. The issue with the grid is that you can see that we're sort of only here. We're just illuminating, say, this bit, and this bit. So we have to shift the grid in order to illuminate the gaps. So we have to do that. So there's a grating shift. So that's now 3 images we have to take. This is only going to improve things in one direction. So we then have to rotate that pattern, and you have to do sort of at least 3 rotations to get a reasonable coverage in all directions and shift. So we end up having to take between 9 and 25 images. If we're working in 3 dimensions, which this technique does work in 3 dimensions, you've got to take quite a few images. But in the end, you get the resolution improvement. So we're down by a factor of 2 over the regular microscope. So this is real, genuine super resolution. And it works in 3D, so we can sort of get a factor of 2 in Z as well. So we've got 2-fold improvement in X, Y, and Z. And just to sort of recap that, we've got some known spatial frequency down here on the bottom one. We multiply it effectively by illuminating the object with that lower one. Those 2 signals beat, and that red curve is generated. So that red bit is the bit that gets through the microscope. So we can basically contrive to get that red stuff all the way to the edge of the regular transmission curve, OTF, and double the resolution of the microscope. So high frequency information is recovered. That's what it looks like in practice. You can do it now. This is a actin and mitochondria. That's a bit better one. So here you really start to see the substructure within the mitochondria. So in the regular confocal, this looks like a continuous sort of smear. We can work out it's a sort of tube-like object. Here we can actually see the detail on the surface of the tube. And then this can do up to 4 channels. So this is salmonella being invading a cell. So in up to 4 channels. What we've got here, yeah, galectin, which actually is the first protein that sort of coats the bacteria when it's recognized, and then a bunch of other ones. So the idea of this experiment was to actually see that these proteins sort of form this sort of onion skin stripe type structure around the bacteria. They're all good so far. We've got this ISIN, which is a variation on the ARIE scan principle. That's faster. We've got 2 ARIE scan systems. They work pretty well. SIM 3 dimensions with some time constraints on how fast it will go, because we have to take a lot of images with various grating rotations and shifts. But in all of these, we're still wavelength dependent, because the microscope itself is still working pretty much as Abbe sort of described it. Getting below 200 nanoliters, that requires something different. So this conceptually can be sort of talked about as a form of switching. And switching itself, we turn something, it's got 2 states, on or off. It's very non-linear, because you basically instantaneously go from being off to on or on to off. There is no partial onness and offness between the two. And that is a sort of sign that something different is going on. Instead, these two different techniques, instead it seeks to turn off fluorescence using optical methods. And then the single molecule localization methods, which are often referred to as STORM, these turn off fluorescence in a chemical way. And by turning stuff off, in principle, you can you've got your extra information, this prior that is required to get more resolution. So let's just concentrate on STED for a minute. That one there. This is how it works. So the STED microscope starts off as a regular confocal microscope. So we've got up here, come on. Well, that blue beam, let's say, is the excitation beam. In a regular confocal microscope, it might be hitting GFP or something like that. Green fluorescence comes out onto the detector and it's raster scanned over the sample in some manner. I mean, in this diagram, they're actually moving the sample around, but we've got to basically focus down to a spot. And that's what that blue dot up the top there, that's our excitation spot. So that's a diffraction limited spot. You know, it's of the order of lambda over 2Na in diameter. Then what you do is you superimpose this donut beam on top of it. And the point at this point is to know that that donut beam itself is a diffraction limited spot. That hole in the middle is also basically lambda over 2 Na in diameter. We're not creating a small, somehow a very small feature up there in the donut. That hole in the middle has got that sort of diameter. But what is very special about it is it's got right in the very center, there is a 0 intensity. So that it sort of fades away till it gets to the center, and then comes up again. And right in the middle, there is 0 intensity. And in principle, that 0 is infinitely small, but basically there's a patch of total darkness, and it is very, very small. The second thing that goes on is this idea of stimulated emission. And that stimulated emission is actually what goes on in a laser. And how it sort of works is this. You've got some fluorescent molecule with ground state, excited state. You excite it with, say, blue light, for example, and it relaxes a little bit, and then would normally fluoresce at some random time later and emit a green photon. If you shine that molecule when it's in the excited state with a wavelength that is in the emission spectrum, so actually matches the band gap, you can sort of have that light going past that excited molecule. The molecule basically senses there's an electromagnetic disturbance in its vicinity, and that stimulates it to radiate. And it radiates exactly at the same wavelength as the stimulating beam and exactly in phase. And so you sort of get an excited molecule, one stimulating beam photon going past, you get 2 photons coming out, and they're matched. And the wavelength is the same, and that's important, which we'll see in a moment. But that molecule is now back down in the ground state. It's been switched off. So this is the switching off process, stimulated emission. And that goes on in the donut. And the wavelength of the donut light is somewhere out here in the tail of the emission spectrum. And what gets through to the image is somewhere over here in the peak of the emission spectrum. But the point is that all that stimulated light comes out in that band, and we can prevent that from getting into the image. Going back to the donut, everything that's under that donut sees stimulation and starts to switch off, except there's a 0 in the middle. So the stuff right in the middle doesn't switch off. So we can just turn up that donut. We can keep on cranking up the brightness, and it doesn't matter how bright that gets, there will still be emission in the middle. But what that's actually doing, in effect, is shrinking down the part that can actually emit. to a very small spot. And here, in this demonstration, they're basically saying that the effect of stimulated emission from that donut shrinks it down to something like 60 odd nanometers, so well below the normal performance of a confocal microscope. And it's really because of this zero there. And the fact that switching off, once you're switched off, you're off, and you can basically turn it up until everything's switched off. But because there is that zero, there is still stuff that's on right there in the middle. And all we need to do to get an image is we know we've got a small spot. We know where our scanner is, so we can get the position of that spot from the scanner with very high precision. We can build up a high precision image from that. So how does this work? So in reality, what's going on is as you turn up the intensity in that donut beam, the spot shrinks and shrinks and shrinks and shrinks, and then it sort of starts to flatten off like this. So the resolution sort of follows this curve. So you get, your first 100 milliwatts of stead beam get you a lot of improvement. The next bit is really, you're starting to really have to blast it to get much more. But it's very easy to get below 100 nanometers. And at some point or other, it does stop working, but 50 is a good number to get to with a well set up instrument. And that's probably all I need to say about this. But there is you can sort of see that there is a penalty here. We are putting in a very, very high power beam here compared to what we would normally use in a confocal microscope. So the normal confocal for normal imaging would be down maybe at 1 milliwatt at the most. So there's a lot of it's asking a lot of the fluorescence, I guess, is the outcome of that. The other thing to consider is that these molecules are getting a lot of excitation and a lot of depletion before they get the chance to actually contribute to the image. So we've got this big sort of 200 nanometer excitation spot, which is then superimposed with the stead donut, which is similar sort of diameter. Anything out here in the wings is getting excited, depleted, excited, depleted, excited, depleted. And it's only when the zero sort of gets to this coordinate does that molecule there have a chance to contribute to the image. So there's multiple excitations and stead depletion before the imaging spot arrives. And that is, this is one of the problems. That's an opportunity for photo bleaching. The other thing is that the smaller the spot, that zero, the fewer fluorophores that it actually contains, therefore the lower signal that we can actually get out of that spot. So labeling density is important. So if you're doing this, we tend to say, so stick your secondary antibodies on a much higher concentration than you would normally, 1 to 50 or 1 to 100 even. How it actually works is sort of shown in here. We start with a regular confocal microscope, this beam path here. So the sort of excitation light comes in and the fluorescent light goes back out to the detection optics somewhere in the confocal scanner. Superimposed on that is this guy here. It's got a wave plate, which basically takes a plane wave and shapes the wavefront in such a way that when it comes to focus, it makes a donut. That's there. So it's really simple. We just have to put a wave plate in there, one component, to generate that donut beam. There's a second thing that we can do. So here you can sort of see the donut is constricted in the XY plane, but it's extended in the Z plane. We can attempt to put a cap on this as well. So if we could put a depletion beam here and a depletion beam here, we could restrict it in Z and make a sort of bottle. You can do that as well with this a pi phase shift phase plate here. Another type of phase plate of a different design produces a focal spot that's got a brightness above and below what we consider the focal planes. And if you combine those two together, we get that. And then fluorescence effectively only is allowed in the center where the zero of that donut is, and in that restricted area in the Z plane as well. So we can basically shut that down in 3 dimensions. So here we're sort of really squeezing the slice of the image, the focal depth of the microscope. So we talked about the confocal microscope last week was producing a depth resolved slice. This thing here basically squeezes that slice down even more. And there's a picture of our STED microscope. That box there is the sort of regular confocal thing. This box here is the sort of thing that generates the donut beams and the sort of 3D cap. And then there's a bunch of lasers in there, and they will just work on the machine as if it's a confocal. So it's really a question of switching on a few extra lasers, and you get super resolution imaging. One of the nice things about the STED is that where the emission comes from is totally determined by where that donut is. Because it's only where the zero, the center of the donut, is where you can get emission from. So if you can contrive to have one STED beam and 2 different fluorescent labels, you can do 2-channel imaging and have perfect overlay of those images. So the channel alignment is built in. That's a very nice feature. So there's some dyes, star orange and star red, that both work with a 775 nanometer STED depletion beam. So that's what I recommend you use, to be quite honest. And that's what it looks like in reality. So there's the sort of confocal resolution of the Golgi in a, I can't remember what it is, probably a HeLa cell or something like that. That's what the STED does. So you can really start to see a lot of fine detail in what look like sort of smooth outlines of these membranous compartments. So we're starting to see the fact that proteins are monotonically distributed around the surface rather than being a sort of continuous film. What about the actual resolution? This is on tubulin, our favorite thing for super resolution. Confocal image, the STED image. You can start to see up the top there, we're resolving 3 different fibrils. There's the overlay, they are the same thing. If we zoom into that and do some cross sections where those white lines are, here's the confocal cross section for the top one. That's what it looks like instead. So we're really getting a massive improvement in resolution. It's resolving 3 different peaks here compared to one big blob up there. What's the full width half max? In this case, we're getting sort of 90 nanometer for the fiber rather than 250 up the top there in diffraction limited confocal image. The actual diameter is a lot smaller, and we'll get onto that in a moment. Here's another example, primary neurons. So here we've just got very dotty imaging of synapses, these structures here. So it's proteins in individual synapses. We can zoom in on those. There's the sort of confocal. We've got a blob here somewhere, presynaptic, and blob probably postsynaptic. PSD, postsynaptic, yeah. Well, the glial one is postsynaptic as well. So they're both postsynaptic actually, but they're there. And instead, we can start to see that there is some sort of domain-like structure of the receptor within the general area of the synapse. This is one of recycling endosomes in 3 colors. There's a confocal image again. These are endosomes, so it's a dotty thing. Zoom in. There's 3 different proteins on an endosome. And when you go to STED, it gets a lot finer detail. So what's going on here is you're starting to see the sorting of proteins within endosomes. That was the general idea there. Okay, down to localization microscopy. Here, we are building up an image fluorescent molecule by fluorescent molecule. That's the general principle of this technique. And we can do that because we're able to switch those molecules on and off. So here's the sort of conceptual idea. Up the top there is a splodge that in this instance consists of 4 fluorescent molecules, all very close to each other. So the problem here is if they're all on at the same time, we just see a larger blob, but we don't really know what's within that. If we were able to switch each of those 4 molecules on one at a time and take an image of it, so here we've got just one molecule on, we can collect an image of it. Next molecule on, we collect an image, and so on. For each of these blobs, our prior information is that this is one fluorescent molecule. So then we can say, well, where is that fluorescent molecule? And to do that, we just really find the centroid of that blob. And we can find that centroid to very high precision, given that prior assumption. We can tell you in XY where this thing is to within sort of 10 nanometers fairly routinely. And then we do that for everything. And then we take those 4 individual images and put them together to get the final output super resolution imaging. So we've got to be careful here with resolution, because localization, knowing where one molecule is not the same as saying, I've got 10 nanometer resolution in my image. So that's a really important point. The density of labeling is an important factor in resolution in this method. So first of all, let's just think about how precisely can you locate one of those blobs? How fine can you sort of put an error bar on its actual position in X and Y? And that depends on the number of photons you detect. So if you detect 10 photons or 100 or 1,000, the error on your sort of XY position changes. And this is in a log graph. So if we assume, and that's actually, this comes from a paper by Thompson, somebody in Webb back in 2002. But that equation there sort of just allows you to calculate, given a number of photons n, what's the error in positioning a molecule in X and Y, and also in Z actually. But the take-home message from this is it's actually proportional to 1 over n, so the square root of the number of photons. So if you want to double the precision, you've got to get 4 times more photons. So there is a price you have to pay. The photon number that you get out does limit the resolution, and it's a 1 over quadratic arrangement. So that's what that black line is. If you have background, it all gets a bit worse. So if you've got a sort of general photon background in your image, the precision goes up that way, gets a bit worse. So, but having a low background is important to get the highest precision for low photon numbers. What is this? So what actually is happening is we're imaging a blob on a camera. This is the regular diffraction imaging spot. It might cover a few pixels, but mathematically we can sort of find the center of mass of that object to within a fraction of a pixel. And then that's just replotted on a much finer grid. So in our image space, we might have sort of 30 nanometer grid and then put that molecule on that grid at an appropriate position. So it's just done computationally, going from a low-res image on a camera to a high-resolution grid in software and on the computer screen. So there's a couple of ways of achieving this. One is by actually having photoactivation or proteins. So photoactivation localization microscopy, known by PALM as the acronym. And these are basically versions of GFP that when they're formed, they basically are dark, they don't emit light. But if you hit them with UV light, typically 405 nanometers in this instance, you can switch some of those guys on. So I can switch on a subset of them, and then I can image that one, and eventually it will actually photobleach. Then I can excite again and start imaging a few other ones. So I'm basically, rather than having all my fluorescent proteins switched on at once, I'm imaging them one by one. And because they don't overlap, I can accurately locate them. The alternative is to have a molecule that changes color. So in this case, we have something like EOS, for example, a fluorescent protein that folds into a green state. If you hit it with UV light, it switches into a red state. And then you do the super resolution imaging on these sparse red molecules, and then just sort of work your way through them. And it may take you 10,000 frames, each with a few sparse molecules, to get a final super res image. The other way is by chemically, sort of a combination of chemically and optical driving. So here you stick all your molecules, the regular labels, something like Alexa 647, and you stick it into a medium that is reducing. So a reducing thiol like beta-mercaptoethanol, MEA, or something like that. And that actually then affects the photochemistry of that molecule. And what you can do is you can excite it. Every so often, it will go to a triplet state and then into a long-lived dark state. And in this, so you're effectively switching them off. But this, getting them into this state here, requires that you stick some chemical in the medium around the cell, around the molecule, which is this reducing thiol. But there you've switched it off. It will spontaneously come back on again by reaction with dissolved oxygen and in the presence of UV light as well. So you can recover them and then do the imaging. So what that actually looks like is this. Here is a bunch of molecules, single molecule, it's Alexa 647 in a reducing thiol environment. They're blinking away. Now what you can see is that they're starting to switch off, right? So the density goes down. If I hit it with UV light, it comes back again. So what's going on is we're sort of exciting here, going round and round that loop, and they're gradually going into this dark state here. So the density of molecules appears to go down, but they're not totally photobleached, because if I add in some UV light, I can get back out of that state into the ground state, and then it's free to fluoresce again. So let's just see that again. So you can sort of see they're sort of going into a dark state, and then if you flash it with some UV light, they will come back again. So that's the sort of general principle of stochastic optical reconstruction microscopy, or STORM. And that's probably the better method to use, because these individual dye labels give you more photons per sort of measurement, which means you get better localization precision from them. That's what it looks like in reality. It's basically a microscope with a whopping great big laser, which is driving them into the dark state, and then getting as many photons out of them as possible without before they bleach. From a technical point of view, because we have to image thousands of frames, single molecule, a few molecules at a time, we have a Z stabilization mechanism in there, and that really helps keep the experiment working. And then there's some drift correction in software, because on the scale of tens of nanometers, things do move over a quarter of an hour or something like that. This is actually our latest version. That's effectively what I just showed you, all squashed into that little box there. But this is something that you can have actually in your own lab. It's portable, so it's up in the Allegretti lab at the moment. But same sort of idea. It's just a sort of state-of-the-art version of a storm microscope. But what it does have is have much bigger lasers, so we can sort of do all that optical switching stuff a lot more quickly. Having more laser power helps you get more channels, to be quite honest, because there's some dyes that work really well, but when you want to do sort of two, three different labels at once, it all becomes a bit more difficult, because the switching, the composition of that reducing buffer effectively needs to be optimized for different molecules. And you can partly get around that by just turning the lasers up. What do you get off it? Here's caveolin. So these are small cup-like objects on the surface of a cell. That's done in with Alexa 647 as that blinking dye. We can zoom in. There's our storm image. So you can already see something's got improved there. We can zoom in further and further, and just keep zooming. At this point, you can start to see that we're resolving some of this, something that does actually start to look cup-like, and that is quite remarkable to me, I have to say. These are individual caveola, and you can just sort of see where we've built the image up from lots of individual spots where we localized molecules and then put them all back together again. And there you can start to see there is clearly some sort of cavity within those objects. And here's the scale bar. That's a 100 nanometer scale bar. So we're way below the diffraction limit of an ordinary microscope with this technique. How to do microtubules, the law for super res. There's a wide field image. That's the sort of corresponding storm image, again with Alexa 647 as the label. There's an interesting point here, is that the resolution of this technique is completely decoupled from the wavelength of emission. The Alexa 647 is actually an infrared dye, or very far red dye. So in principle, if we in the realm of structured illumination, a very poor choice. This one, when we're doing localization, we don't care what the wavelength is. We just want to know where it is. And that depends on the photon budget. And we can zoom in on that, and you can see sort of fine detail. Again, we do the characteristic line profile to see what's actually going on. We've got 70 nanometers again. So here we start to see the disconnect between localization precision, so how well I can say put an XY coordinate on one single dot, and then the structure as a whole comes out as something different, right? It's coming out as a much bigger number. So this is speaking to the labeling density. And just as an aside, we can introduce an astigmatic element into this system, which means that the focal spot changes shape depending on where you are in Z. If you're a bit above the focal plane, it sort of elongates in this direction with the cylindrical lens, and if you're below, it goes a bit elongated in the other direction. So this distortion here is encoding depth as well. So in the sort of plus or minus a few hundred nanometers in this case for this example. So we can basically get that image in XY and then add depth information to it. So in this case, the color scale is a depth code. So we can basically see this filament was lower than that filament and stuff like that. And that's an easy addition to these sorts of microscopes. Here's an example in two colors. So now we're using City Fluor 568 in one channel and Alexa 647 in the other, these cells have been irradiated and the proteins are starting to cluster. So this is a radiation-induced focus somewhere in the nucleus. I'm not at all familiar with what these proteins are, but the point is we go from this diffraction-limited splodge up the top here to actually see the distribution of those individual proteins within those foci. So how well do these systems actually work? Here's some data from the Gregor group. Alexandra who was previously a postdoc in this group, was looking at synapses again. So here's a sort of synaptic structure here, or imaged in storm microscopy. What we really ask is how densely was that thing labeled, and what does that mean for resolution? So first of all, the actual individual localization, then we sort of just histogram the accuracy, the precision of localization of all these individual molecules as they came in. We're getting sort of numbers around 10 nanometers for actual localization precision themselves. And in Z, it was down to about 20 for this particular sample. All good. Jerome did all that. The question is, what's the actual resolution in the image? For that, we're borrowing a technique that's used in electron microscopy with sort of Fourier shell correlation, or Fourier. And really what you do is you take the data set, split it into two, and you overlay it. And then you ask how far do I shift one image relative to the other for the correlation function to drop off to some threshold value. So we've got that image. We can split it up because we've got lots of individual molecule localizations. We can just put them into two piles, if you like, and then form the image and then do a correlation function, which is basically overlaying them and then moving them apart and seeing how far you go before the correlation function drops off. And that's what you get from here. There's a sort of spatial frequency cutoff, which, if you invert that, you get a resolution of around 60 or 70 nanometers for that image. You can plot that as a map, actually. So you can divide the image up and just sort of see where are the high resolution parts. So the higher resolution parts are here. This is outside the synapse, actually. All this dark stuff is zero. There's no signal there. But the darker ones of these pixels are sort of down here on this scale, below 50. And then there's some patches where the density of labeling is low and then the resolution is much less. So we can sort of inspect, we can basically have a sort of analytical tool for looking at resolution in reality. The other thing to consider is our labels. So most of what we've been doing so far is all antibody labels. So we have this toolbox of fluorescent stuff, antibodies. Well, structural biology has come on a bit since that squiggle that I did there, but they're pretty big, right? They are of themselves a good 10 nanometers in diameter. So if we're attaching one of these onto our protein of interest, we're sort of adding a 10 nanometer spacer onto it. Things do get better as you go down. So the GFP itself in Palm, that's of the order of 3 or 5 nanometers as a spacer, if you like. So there's an ambiguity there. Single molecules themselves, the fluorescent molecules themselves, are down at sort of 1 nanometer. So, Alexa 647 is that sort of dimension. So that's the sort of errors with labeling. With primary and secondary antibodies, we're basically doubling up, right? We've got primary against tubulin, then a secondary, that's sort of another 10, and then our fluorescent molecules on top of that. We're sort of adding 20 nanometers with that on one side, 20 nanometers on the other side, and then we get sort of the structure itself is 25, and then we end up at sort of 65 nanometers wide. So that's the general explanation as to why our tubulin images don't come out at 25. It's just the physical size of the labels. So the way around that is to use much smaller fragments. So these are typically used like nanobodies or FV and Fab fragments, so smaller parts of antibodies, get you a significant improvement in resolution just because of their smaller size. So we recommend you sort of go onto those for your labeling, and also think about doing primary labeling. And the other thing is labeling density, again, which was just coming back to this idea that if that's our object, we decorate it with a few dots of fluorescent molecules, you can't really make out what it is, but as you increase the density of labeling, it becomes apparent what it is you're actually looking at. That's the other side of localization. You really need to make sure you have a sufficient density of labeling to see the underlying structure. So in the end, we're just summing up now. We've got these sort of three different classes of SIM, structured illumination microscopy, STED, and localization microscopies, Palm or Storm. You can sort of see the resolution that you can get out of them. Factors of 2 with the structured illumination, basically in XYZ, and maybe a factor of 10, possibly on a very good day with localization. But again, that's limited by the density of labeling in reality. It's easy to do 4 colors in structured illumination. I mean, that's just regular confocal microscopy, regular fluorescence microscopy, but just using patterned illumination. We can do 3 colors, but actually what starts to get us is the photo bleaching. But what is really nice about that is there's really no post-processing. This is the super resolution image comes off the microscope in one go. In SIM, in its finest form, there are sort of 9 to 21 images to maybe 25. If you do lots of rotations, you've got to do some image processing to get your image out of that. Then from a practical point of view, you've really got to start at the diffraction limit and prepare your sample well. That means sort of high tolerance cover glasses, which basically are 170 microns thick, because that's what their objectives are designed for. No phenol red and stuff like that to get low autofluorescence and low fluorescence background, because that just eats away at resolution. Refractive index match, because most of these microscopes are designed around oil immersion objectives to start with the highest NA. If you're immunostaining, increase your antibody concentrations to get a high labeling density. And what we've got, these are the systems we have in-house. Can these be improved? I mean, there is really a sort of next generation instrument which uses a variation, sort of an amalgam of STED and localization microscopy, which sort of gets you down to 3 nanometer resolution in the localization. It's a very expensive machine. So there is one at Harwell, and you can basically sign up for instrument time on that if you want to go and use it. But what you would want to do is basically perfect your imaging on one of our in-house instruments before you took a sample to the MinFlux system, say. And some acknowledgements. Light Microscopy Facility, Jerome, Ben Sutcliffe and John Howe, who are former members, James, who's still here, Agnes from the Roundhouse lab for the bacterial work, Jake Watson and Alex Pingera from the Gregor lab, Sophia Brusium from the Seaman lab in CIMR, and Lena Haas from MRC Cancer Unit. Right, that's it.